Hello, I'm Michael Keenan, Conference Director of TIDES. I'm here in Long Beach, California, with over 850 attendees from 30 countries around the world. TIDES covers the latest developments in oligonucleotide and peptide therapeutics, from discovery all the way through to manufacturing. Some of our speakers this week have agreed to share their insights with you on the development of oligonucleotide and peptide therapeutics. Please enjoy the presentations.
Well, I'm Danny Zurr. I'm the founder and the CEO of Quark Pharmaceuticals, and I'm glad to be here at TIDES, and I'll be happy to answer some questions. Well, you need a lot of perseverance. Everything takes time. It's like developing any other drug. There are particular things that is more related to nucleic acid compared to antibodies or to small molecules. But basically it's drug development, which is capital intensive and takes a long time. Well, there are some difference between developing sRNA or nucleic acid to other things, and I can point out several of them. I mean, for example, one of the big differences that most of the small molecules, and sRNA is considered a small molecule, they are promiscuous. They're targeted a certain receptor or a certain gene or a certain, but basically they have all other things. sRNA, on the other hand, or nucleic acid, like antisense, they are very precise. They go directly into specific target. And that creates several problems. I mean, one of the problems, for example, in development, that when you prepare a nucleic acid like sRNA to target a certain gene in human, and you want to do some animal studies, it's not necessarily means that this target, the animal studies, have a different genome. So you have to prepare, something, you have to do more work in tox studies, preparing IND, because you have to treat the animal both with a real drug that you want to develop, which fit human, and then with drugs or molecules that actually fit the small animal or big animal. So it creates more problems. Also, very often the sRNA, particularly because that's what we are doing, is not toxic. And very often you have to give a huge quantities into the animal before you see any symptom of NOEL or things like that. That's some of the problems, but it's drug development. Well, India and China, we were the first company, basically, that got approval for using sRNA in clinical studies, in pivotal study, basically. It was an interesting experience because they for the Indian and for the Chinese, it was first time. They were not aware of a lot of things that in the West, in Europe and US, I mean, there are a lot of clinical studies. Even the fact that it should be considered as a small molecule and not biologic was something that we have to basically to convince them. But it was an interesting experience. It's not easy. It's very difficult. But we are there, and hopefully we pave the way to other companies to come to those markets. I don't have to tell you that from clinical study, there are a lot of population. I mean, it's the biggest population in the world in those countries. They have very often very high standard of care in hospitals, and a lot of the principal investigators spend time in the West, and they basically trained, and they just came home actually to continue work at home. That's Well, there was one breakthrough that we were involved. We received the approval from the US FDA to use sRNA systemically in the body. Up to that, this was like 4 or 5 years ago. This was a breakthrough. Now more and more people are using systemically. Obviously, one of the key things in siRNA is delivery to a certain target. There are several small breakthroughs that may be additional major breakthrough. We know how to deliver it to many different organs, but there's still a way how to improve it. And I think that the first drug that actually will get an NDA and hit the market, then it will be a big breakthrough, and everybody recognizes that this is a legitimate category of drugs. I mean, that's basically it. That's the next important breakthrough. Maybe in 1-2 years from now. Yeah. Look, the best thing is to draw some analogy from the antibodies. It took like 20 years or more from the discovery of monoclonal antibody since the first drug actually hit the market. And it's a huge market today. And that's what happened with siRNA, no doubt in my mind. 5 years from now, there will be several NDAs approved, and everybody will move forward actually to develop drugs based on siRNA. So it will be a huge market, there's no doubt about it.